Okay, so today we have a discussion session. What I'd like you to do, I want to make the grading part of this really easy. So just, I'm not going to take up the whole time today. If you guys want to chat afterwards and either give me something on paper or email it to me or post it online, any of those are fine, okay? Lighting is terrible. Is it this thing? There we go. How many of you guys know this artist? MC Escher. He's my favorite. So this paper, The Influence of Sex-Handedness and Washing on the Diversity of Hand Surface Bacteria, by Noah and Rob Knight's lab. Same folks as we talked about the other day. This paper is actually a little earlier. This is from 2008. So, the question is, what is normal for hand flora? And how much variation is there? I mean, are your two hands the same? Are your hands the same as your neighbors? All this kind of stuff. Are there any consistencies in hand flora? This is important because people are really keen to disinfect their hand these days, right? Especially flu season and this sort of thing. Hands are vectors for a lot of disease. Now the question is, what's the microbiology going on there? And basically, we don't know. And we've seen this before. So there's a question, which we're talking about in the regular part of this class a lot, is how do you take a census of microbial diversity? It seems like a very basic sort of a question, but there are a lot of approaches, none of which are ideal. Number one is cultivation. And we've seen in lab, and we've talked about the fact that this just doesn't give you a very representative picture of what's in an environment. Ribosomal sequence surveys, cloning, sequencing, and looking to see who's there. This is an old-fashioned sequencing, John. Nobody does this anymore. And yet I've got boxes of them in the lab. Yes, you did have to go through GATC and read these one off at a time. CT, CT, and so forth, and manually read these on. Denaturing gradient gel electrophoresis. This is actually taken from the, no, actually it's not from the book. These are more interesting populations. You can see the kind of diversity that's in these populations compared to these. TRFLP. This is from the lecture last time. Fish. This is a nice image. This is using 3 different probes with 3 different labels. And you can see you can just do a nice census and look at who's there. This particular one, by the way, this is false color. It's not the real coloration. But there's a universal probe and then 2 phylum-specific probes. We're not going to talk about phylogenetic arrays because this approach, although it was very promising, just hasn't ever developed it yet. So we talked about how you can use PCR primers to target particular phylogenetic groups. Now imagine taking those oligonucleotides and printing them on arrays and then just washing genomic DNA from an environment over those and seeing how much signal you get it on each spot. You can do that and you get nice arrays like this, and you can do 2 colors and look at differences in populations. But we saw in the paper on chloroflexus from wastewater that group-specific probes are hard to come by. They hit some members of the group and not others, and this sort of thing. And so interpretation of this kind of data has turned out to be a computational nightmare. And so I don't know anybody who's doing this. The approach we're using today is ribosomal survey analysis, and in particular using 454 sequencing to get lots and lots of sequences at once. And this is the ribosomal RNA. This is exactly the same set of primers that we talked about with the human microbiome. And so you have a 27 forward sequence, a 338 reverse. You guys know how these things are named? Primers for PCR and things like this? So when I talk about 515 or 1492 or 27 or 338, those are the numbers. This is the E. coli ribosomal RNA, which is typically used as a standard. And the 5' nucleotide on this primer is nucleotide 27 in the ribosomal sequence. And so that's where these numbers. 515 is right here. 1492 is down in here somewhere. 338. etc. These things are barcoded, so you can use, you can do lots of experiments and get all the sequences in one shot. They are, once again, error-correcting barcodes, and I take a little bit of a different perspective on that than Rob does. He's called these error-correcting barcodes, but they're not really error-correcting, they're error detection. You can't correct any errors with these barcodes, but if there's a problem with the sequence, you know it because the barcode doesn't come out right, and you can throw that sequence away. The complexity of the setup of these kind of experiments takes a little getting used to. So you've got basically two primers. You've got this primer, which is constant for all of them, and then a primer here that's got this stuff and this stuff you need for sequencing, this stuff you need for priming, and a barcode. But for every experiment, for every individual and every sample you take, you have to have a different barcode, which means you have to have a different oligonucleotide. And so these are the barcodes they use just in this one paper. And so in terms of primers, each one of these has the ribosomal priming sequence here, the barcode, and then the sequencing sequence over there. So this is a bunch of oligosynthesis, and it's a bunch of keeping stuff straight and logistics and setting up reactions and making sure you're doing everything right. The logistics of this kind of experiment really are tough. So what's the data? They did 27 men and 24 women, both hands. And they just swabbed the hand. They didn't do subparts of the hand or anything like that. This is one experiment. This is actually not an entire 454 plate. They did two other experiments on the same plate. This is just one of the three experiments they did. And so they've got 351,000 sequences. They got over 1 million on that one plate. Average length of 228 nucleotides. They got 331,000 classifiable bacterial sequences. Not all the sequences they got were sequences that they could interpret. So they would get a sequence, and it's so different from any other bacterial sequence that they don't know what to do with it. And so they have to be removed from the analysis. Number of phylotypes. So once they've got a sequence and they can interpret it, what bin does it drop into? And phylotypes can be at all levels. I mean, in some cases, you'll know what species it is. In some cases, you'll know what genus it is, but not what species, and so forth up the taxonomic chain. If it can be dropped into any phylogenetic bin, then it's in this list right here. This is almost exactly, by coincidence, the number of characterized and named bacterial species. It's about 5,000. They detect that many, on average, on one hand. All right? Think about that. Average, in some cases, in some hands, they add more sequences they detected on one person, one hand, than all the bacterial species that have been characterized and named. Average number of phylotypes per hand. Excuse me, phylotypes. On average, you have about 158 sequences per hand. I misspoke. They got about 3,000 sequences per hand. But when you add them up, they come up to 5,000.
So we have like anthrax in our hands?
Sure. In some number, yeah. Yeah. That's right. So they use this thing unifrac again. We talked about unifrac before. It's basically a way of taking two trees, superimposing them, and looking at the unique amount of branch length in one tree versus the other. You don't have to worry too much about how the numbers are done on this. Basically, so the average number of phylotypes per hand is 158. Of those 158, on average, 21 of those are shared between two different individuals, like the right hand of person X and the right hand of person Y. That's kind of remarkable, don't you think? Because that means that most people have most of the organisms on your hand are unique when you compare them to one other person. Now, on the other hand, it's true that these 21 are usually the same ones from person to person and are the predominant organisms numerically on your hands. It's the few percent of outliers that are variable from person to person. This is 30% similarity, and you're more similar between the two hands of the same person, left and right. So on average, 28 of the 158 unique sequences on your hands are the same between your two hands. So not surprisingly, the two hands of one person are more similar than two hands of two different people. So this is, again, this unifrac distance, and so this means more similar, this means more different. How this is actually calculated in this context and treated out is a little kind of weird. Basically, this 2423.23 represents variation within the average variation across the group, and that's why they all end here. The first thing to notice is that male and female hands are readily distinguishable. No problem. And I'll show you how that is in just a minute. In other words, if you take a swab from someone or a bunch of people and test them in this way, you can readily bin them out and identify male versus female. You can distinguish dominant versus non-dominant hand. And so again, you could take that swab from an unknown source and say this person, that swab was taken from their dominant hand. Now you can't tell whether it's right or left. You can only tell whether it's dominant or non-dominant. You can also tell how long ago it's been since they washed their hands. They don't test anything more than 4 hours. I think it would be interesting to go further out than that and see whether that makes a difference or not. You can combine these things. Now, this is what I find really interesting. Not only can you determine whether it's male or female, dominant or non-dominant, and how long ago they washed their hands, but you can tell that it's a female who washed her hands less than 2 hours ago, and it's the dominant hand. Whoops. All from one set of sequences extracted from that. This is really interesting. So this is male versus female and how long ago they washed their hands. Male and female hands are most similar right after they're washed. And then, within 2 hours, separate out and become more similar based on gender rather than how long ago they washed their hands. So in other words, your hands, when you wash them, you homogenize them a little bit between sexes. And then pretty soon after that, they become more distinct. Female hands harbor more microbial diversity than male hands. This actually created some headlines. I don't think this is a big deal, but it raised headlines, things along the lines of, female hands are more hospitable to deadly microbes and all this kind of stuff. When newspaper people get a hold of things, sometimes the science goes by the wayside, I'm afraid. And so these are two different diversity metrics, and these are saturation curves. We looked at these before. So the way you interpret these, this is average number of phylotypes per hand versus the number of sequences that you randomly extract from the database. And so male hands can show fewer numbers of sequences per any sample size than female hands. And there's a nice statistical difference between these. There's no doubt about that. And this is the number of phylotypes per hand. Phylodiversity. The first one is phylodiversity. In other words, if you take all the sequences from the hand, generate a tree, and add up all the branch length, that's phylodiversity. The reason that's important, by the way, let me pause for that. If you've got 5 sequences, which is more diverse? This or that? These are more diverse, right? And so the way you do the phyloδιversity, one way of doing that is just to take the tree and add up all the branch length. And so if you've got, this sort of a thing, you can add them all up, and the sequences are all similar to each other, contribute to diversity, but not very much compared to the lone branches. This is just adding up the number of unique phylotypes. Oh, let's see. The level of diversity between hands is usually similar. In other words, for any particular person, if one hand is highly diverse, then the other hand probably is too. This is no surprise, right? And so these are individuals. Individual 1 here has low diversity on their hands, right and left, 2 and 3. Why might one person's hands be more diverse than another's? Occupation would be a big one. If you work on cars and spend your time with your hands in gasoline, how diverse is your hand microbe going to be? Probably not so much, right? If you, unfortunately, work in a hospital, how diverse is your hand population likely to be? Yeah?
What about, because they do these types of, couples? See if their hands...
That's an interesting question. I don't know the answer to that.
I know that they said that you could find the same, mouth flora of couples.
Yeah.
I'm wondering if you could find the same, diversity among hands among
Yeah, so people talk a lot about how at least gut flora, and probably more than that, are inherited non-genetically from mother to offspring. But couples obviously could do that as well. In the case of STDs, very directly. Level of diversity per hand doesn't change significantly with washing. Washing is futile when it comes to the normal kind of organisms on your hands. All right.
What's the point?
The point doesn't have to do with bacteria. The point has to do primarily with viruses. Now, you can do this kind of experiment with viruses. You can't use ribosomal sequences, and you have to target particular groups of viruses one at a time, because you don't have primers that'll hit, T4 and POX virus at the same time. But you could do this with viruses.
Has it been done?
Not that I'm aware of.
So washing is more effective against combating viruses?
Against viruses, apparently the bacteria glue themselves on and aren't likely to come off. In terms of diversity, now the numbers of organisms will go away, will go down. But the different kinds of creatures, the spectrum of creatures on your hands doesn't change. And obviously, you want to get the crud off your hands. Not just bacteria, but other stuff.
So what's your opinion on hand sanitizer?
I don't like it.
Okay.
No, I don't. And I don't like it for a very specific reason. A lot of them are alcohol-based. And the primary way that you prevent disease is not through antibodies and T cells and that kind of stuff, but just a physical barrier. And alcohol is not good for that physical barrier. That being said, I do wash my hands. There's no difference in diversity between dominant and non-dominant hand. I said differently last time because in one of their experiments on that paper, they mentioned this. But on their actual data they show here, they are not able to show any statistically significant difference in the diversity between the two hands. Now, the organisms that are there, we've already shown are different. But in terms of the total diversity, the number of unique phylotypes in this particular case, there's no different. So even though your hands have different flora from hand to hand, one of them isn't much more diverse than the other. Let's see, what's this? Not only are females more diverse than males per hand, they're more diverse per individual. In other words, if you add up the diversity of the two hands, females also have more diversity. This was a concern. This isn't necessarily the case. You can imagine that if females had more diversity per hand, but their hands were more similar to each other, that males could have more diversity overall, but not per hand. But that's not the case. diversity is not higher per hand, but it's more diverse per individual right after washing. I find this really surprising. In other words, when you wash your hands, the diversity of organisms in your hand goes up, not down. What I take this to mean is that you're not washing off different kinds of organisms, you're washing off the high concentration organisms. And so when you add up the diversity after that, you end up with more diversity. Hands are most different from each other right after washing and then quickly become more alike. This makes some sense, right? Because once you wash your hands, you're likely to touch them together and transfer organisms between the two of them. What I find interesting is that they are fairly diverse after washing. And what that means, I'm not clear on. Who are they? Primarily Propioniobacteria. This is the big one. Anaerobic, gram-positive bacteria. They make propionic acid. This is the kind of body odor smell. That's not too surprising, I don't think. And if you do cultures of people's hands and incubate the plates anaerobically, this is something you can get a lot of. Who else? Streptococcus. That's no surprise, right? Staph, primarily Staph epidermidis, by the way, but also Staph aureus and everything else. Corynebacterium. I'll show you later. This is primarily males. Burkholderia, Moraxella, Lactobacillus, primarily females, and so forth and up the line. And then a bunch of so-called others. These are things like chloroplast, pollen, thermos, and so forth. I'll show those later. The danger here, and I point this out very carefully, is that anytime you're doing ribosomal PCR, you have to worry about how good your primers are at hitting everything. Because these numbers all depend fundamentally on how evenly your PCR primers amplify things. And never is that as good as you hope it is. So here's the breakdown. Male versus female, for example. What is it? Males have a little more Propioniobacteria, probably not statistically significant. Males have a little more Streptococcus, which is statistically significant. Females, lots of Lactobacillus. Males, a little bit more Corynebacterium, more Pseudomonas. This makes sense, right? Lactobacilli are commensalistic organisms of the female parts and Corynebacterium of the male. And, you're all one body, right? This stuff gets around. How quickly it gets around, I guess, is up to you. After you wash your hands, you'll see that the concentration or the prevalence of Propionibacteria goes down. So Propionibacteria is the major thing you wash off when you wash your hands. But it's a normal organism. It's the predominant creature there. Should have brought my ruler today. Staph becomes more prevalent right after you wash your hands. This is an artifact of the way they do this data. This is percent. And so if you wash off a significant fraction of the single most abundant organism, the relative percent of everything else will go up, right? And so that doesn't mean that when you wash your hands, you're applying Staphylococcus to your fingers. It just means that because it doesn't wash away as well as the Propionibacteria does, it becomes a larger percent. Likewise, Burkholderia, Moraxella, Corynebacterium, and so forth. Not a lot here I don't think, except for the Propionibacteria being the major thing that you're washing off your hands bacterial-wise. These are the numbers, and there's tables and tables and tables of this stuff in their data. What are some interesting ones in here? Lots of mouth organisms. People bite their nails and all that kind of stuff, right? Touch their lips. There's one here I found really interesting. Thermos. And there's more thermos on your hands right after you wash them than any other time. Any ideas? Thermos grows at hot water heaters. What is a hot water heater? It's a big continuous flow fermenter, right? Groundwater comes in, it gets heated up, it's held there, it gets trickled out, new water comes in. It's a continuous flow fermenter for organisms that grow at those temperatures. And it's one of the things, I used to do this in Diversity Lab, and I think we'll pick it back up again, is ask people to bring in, you're supposed to drain your hot water heater once a year to get the crud out of it. This settles down to the bottom, and nobody ever does it. So if you drain some of that stuff out, it comes out with rust and stuff in it. And if you just streak that out onto an LB plate and incubate it at 65 degrees, you get thermos everywhere. Hot water has thermos in it. And you apply this to your hands every time you wash with hot water. Now, thermos is not a pathogen, obviously. It gets on your hand and it's in the deep freeze, right? So it's nothing to worry about, but there it is.
But it can't grow very well on that.
It can't grow at all at 37 degrees. There's some gammatids in there. All kinds of interesting stuff. Occasional chloroflexi. This is a good case that shows you that basically, if you sequenced, instead of sequencing a few 100,000, if you sequenced a few 100 million, you could probably detect anything at some level, right? Those extinction curves I showed you don't plateau anywhere in the near future. They continue more or less on indefinitely. So what's all this mean? Propionibacteria are the predominant organisms of your hands, and then the other organisms that you already think about as skin microbes make up most of the rest. Strep, staph, Corynebacterium, Burkholderia, Moraxella. You guys know what Moraxella is, right? It's a common symbiont of your eyes. Lactobacillus, Pseudomonas. I mean, none of these things are a big surprise. There's large variation between the hand microflora, between males and females, between how long you've washed your hands, between individuals, and even between the two hands of the same individual. Now, if you go back to this, it's not the propionibacteria that are any different, right? And it's probably not even these things down here. It's these organisms in the other category that are the primary differences, except between sexes. This is where most of the diversity is, right? This is most of the organisms, but this is where most of the diversity is, because this whole other is primarily a category of one or two sequences collected, and that would be very different from person to person. Defining normal flora is tough, right? If you take a diseased person, they're still going to be primarily propionibacteria on their hands. And there's going to be staph and strep and all that kind of stuff. So the question is, what is normal? I think we can determine that. I don't think we know how to do it yet, but I think that we'll be able to determine that. And it's not going to be in the kind of gross, there's MRSA kind of thing. It's going to be by looking in detail at the numbers and the relationships between numbers to see what's going on. Quantitation of this kind of stuff is absolutely the key. And so, if you take a look at this hand, I think you're hard-pressed to find anything here that's wrong with it if you look at it in detail. Look at the nuts and bolts. Everything fits together correctly. It's only when you count things that you realize that there's a problem. This person has polydactyly. And that's it. What's that?
I didn't think it has like the second alphabet of it.
Yeah. So what do you guys think?
Definitely makes you start thinking about your hands and washing your hands like this.
Yeah, the statistics for hand washing are pretty abysmal. Well, yeah. So, obviously, you've got to wash your hands to get, here I was this morning scraping drywall and you cut yourself, you've got to wash your hands, right? You've got stuff on your hands. You get grease and dirt on your hands. That's got to happen. Certainly if you're in a hospital setting, yeah, you sneeze and stuff, you get viruses on your hands. That's important. But you're not going to change the normal flora on your hands, and I don't think you want to. I think this whole notion of antibiotics and everything, and antimicrobial baby toys and this sort of thing, is seriously misguided. No, I'm not kidding. They put stuff in baby toys so when they chew on them, they're not getting them out little organisms. That is such a bad idea.
But as soon as the baby touches the toy, won't it?
Well, so they get stuff on it, and then the antimicrobials kill them,
So then the microbial don't disappear after a certain time.
and the baby ingests them, and it's a bad idea. Yeah.
What's the significance of, like, you ever heard of, like, microban? Like, they put on, like, shapes and other things, so, like, I guess it's really
Yeah. So, I don't know the answer to that particular one. The issue with your feet, there's a lot of propionibacteria on your feet, and that's because it's a closed humid environment. When you wear shoes, that gets kind of nasty. But it's typically fungi that are the problem with your feet, and that the microban kind of stuff doesn't do anything for that. Yeah.
When they did the samples for this, did they take a where on the hand? Like, did they do it under the nail or anything? Because sometimes that might be different than.
That they would. No, what they did is they took the palm and swabbed it over the entire whatever surface this is. I don't know if that's ventral or dorsal. I think it's going to be ventral. But they take the whole surface and swab it. They didn't do the back of the hand. They didn't do between the fingers or under the nails. I don't have any nails. That would make a big... And it would be really interesting to look in more detail about where stuff is. Yeah.
Studies like this make people with OCD, things like that, go crazy.
Yep.
Because they have you don't realize how much microbes are on your body. Obviously, you can't see them, but if I had germaphobe problem, I'd freak out if I saw this.
Absolutely. Well, it's like the bed bug thing, right? Insects and bugs bother people.
Those
And-
are quasi-visible, though. You
Well-
can't tell that you have.
Well, okay, but you can do an experiment in lab sometime when you have a chance, is to get a really fine, like a mascara comb kind of thing, run it through your eyelashes, eyebrows, and make a wet mount, and you will find arthropods in there. Yeah.
How would you protect, what the condition is, and if you manage to eliminate all microbes?
Oh, interesting question. So this is the whole thing about what microbes are good for. The obvious thing is that if you've got a good coating of propionibacteria and staphylococcus and that kind of stuff, you're less likely to have problematic organisms. An organism like MRSA is going to have a rougher time getting a foothold on your hand if you already have non-problematic staphylococcus there, because it has to compete with those. But in terms of what if you eliminated everything, would there still be an advantage? And I think in the case of your hands, no, I think that's unclear. I don't think anybody knows. In the case of your gut, there are very clear advantages to the microbial population of your gut.
Can you technically clear your hand of all microbes?
You would have to do that in the same way that they create notobiotic mice and that sort of thing. I don't think you could do it with your hand. I mean, so surgeons, of course, will scrub and scrub and scrub and do all that kind of stuff. Their hands are not sterile. But they've removed the problematic kinds of creatures, or at least hopefully have. The big thing with hands, the doctors in their hands, you've heard the story of Jenner, right? So, yeah, Alan.
Are there alternatives to alcohol sanitizer?
Yeah, there are. There's some that just have detergents and that kind of stuff, and those are probably better. I don't know. Soap and water is probably better. Get a good coating and throw it somewhere. Yeah.
Clearly, I'm an avid hand sanitizer user.
Okay.
So,
And so what happens is, see, my hands are dry, and if you use hand sanitizers, it dries them out, and you get cracking, bleeding, and
oh, I definitely do.
yeah, and so you're exposing yourself to more potential infection that way than you ever could by just the stuff that normally gets on your hands.
I don't know where I heard this. I don't know if it was Dr. Kitty or Dr. Oz. I don't know. I heard somewhere that if your hands are clearly dirty, then the soap and water is obviously your best choice. But if they're, like if I wanted to go eat now, my best option would be the alcohol based.
That, I think, would depend on your goal, right? on the other hand, are the microbes there really a problem? I mean, if they're on your hands, you're ingesting them one way or another anyway. I think the issue with at least alcohol-based hand sanitizers is that you're probably doing yourself more harm every time you do that. I wouldn't use alcohol-based hand sanitizers. Yeah, just go in and wash them.
Sometimes it's just more convenient.
They're built into the cafeteria.
Yeah, they're everywhere. They're everywhere.
Do you think they'd work though? Because I know a bunch of people say, well, oh, you're just building immunity.
Well, I think in terms of the bacteria, which is what this paper is about, there's no evidence that washing your hands helps in any
Helps
way.
them.
That way.
Okay.
But in terms of viruses, there's clear evidence, particularly for flu virus and that sort of thing, that hand sanitizers do remove those viruses or eradicate them or whatever you want to call it. Yeah, Andre.
So even though big fat liars when they say that they kill 99.9%?
No, it's not true. It does kill. Yeah, it's a logarithm thing. So you go from 100 billion to only 1 billion. That's 99.9% dead. And what they're talking about isn't all the organisms that are actually there. It's only removing the ones that are removable through a swab of some kind.
Or do they put that on the package?
No.
So it's false then?
And it's worse than that because what they actually measure are plate counts. So none of the propionibacteria show up because they don't incubate them anaerobically.
They're a little sneaky then.
Yeah.
I've seen it. It was really odd. I was flying and it was a surgeon. He wore gloves, like surgical gloves, all the time because he didn't want to touch things. It seems weird. But would that though facilitate? Because they're really tight on him, and you can tell he's been wearing them for a long time. And he had sushi in his bag, whatever. It's really creepy. But wouldn't that obviously facilitate more anaerobic?
I would think so.
Bacteria.
I would think so, but I don't really know. I saw a funny thing once. This was before they renovated what's now called Thomas Hall. There was a side entrance there that people used to go out and smoke. And so here's this guy that obviously is a lab tech. He's got his jacket on and his blue gloves, and he's smoking a cigarette. I'm thinking, okay, the gloves are designed to protect what from what?
Yeah.
Not to get the nicotine on his hands, not to get organisms from his fingers onto the cigarette which he's smoking. I don't know what it was.
You didn't ask.
That's not the sort of discussion you're likely to want to get into. Well, so here's a similar but a very worst case. I used to have to go to the cancer center over at Duke quite a lot, and it's dismaying to see on the front entrance of the Lineberger Cancer Center a collection of people in wheelchairs with bags and them and their nurses out there smoking cigarettes. That is, I don't know.
Well, in that case, you just really can't feel sorry for them.
I think you can feel sorry for them. I don't think you can understand them. But anything else? Oh, yeah.
So this might be a really odd or stupid question, but how would you have anaerobic bacteria growing on your hands that are always exposed to air?
Yeah, it's an interesting question. The reason is because although your hands are aerobic on the outside, if you look at them microscopically, there are crevices and pores and all this kind of stuff which make up the predominant part of the actual environment. When you think about how surface area works, most of the surface area is in these little cracks, crevices, nooks, crannies, and all that kind of stuff, and that's where the organisms are. Yeah.
I think it'd be an interesting study to do babies' hands.
Yeah.
People who are like construction workers with calluses to see how well the type of microbes, obviously babies, but also too do the calluses and the roughness of the hand, does it certain microbes like that?
There's a whole bunch of interesting stuff there. See, from my perspective, what I see interesting is when a baby is born, they are presumably sterile. So this is an ecological question. How is a normal flora of the hand established? presumably there's an ecological succession, just like any other new ecosystem. People have looked at that in terms of teeth and the microbial succession that takes place when a tooth erupts. But what about a baby in the first place? They've looked in the gut, they've looked at teeth, but I don't think they've looked at the skin or any of the other concepts
Well,
like
like
that.
if you just touch your mother's hand, you get those microbes, but if
Right.
you pass that way.
But does it, typically you wouldn't expect that these organisms would be able to get a foothold on a completely sterile surface. And so presumably there's some organisms that make landfall and establish themselves, like beach grass, right? And then you ultimately end up developing a mature ecosystem. All right? So don't forget to post a comment or question or thought or something on the website, or email it to me, or hand it to me, or whatever. And those of you who are in lab, I'll see you at 1:30. Yes. Yes. 13 discussion series and a lot of things to try. So if you've got potential already, you don't want to confirm it, you're done.
They say for like babies, or like placenta mothers. If anything, that could be what they could feel like placenta.
What's that?
Like I've never done anything like mother's placenta, and there's microbes.
You don't cultivate that. You take serum and PCR. Particularly people that have and the plaques are full of organisms that are normally associated with dental.
Like endocarditis and stuff like that.
Exactly. Alan, yeah, what's up?
Yeah.
Like 100 million are we essentially their carbon and nitrogen source? Or, like, I know there's gotta be some, like, cross-feeding.
Right, certainly a lot of them are leaning on each other. It's a complicated ecosystem and so forth. Yeah, and so the question is where is all that stuff entering into that food chain? And presumably not a lot of it is autotrophic. You might expect some photosynthesis on your body, but probably not a significant amount. And so yes, ultimately either you or the stuff that flows over you is a food source.
Do they no big component of dust
So like that's
and how this is mainly skin cells?
and then white.
Did they, or can they compare, like, somebody in a room?
That's an interesting question. So as far as I know, that hasn't been done. I mean, it is straightforward to take dust and extract human DNA and identify, they've got a bunch of them. So I don't know how much